Hello, everyone. I'm Aryan Kamal, PhD student from Zog Group at EMBL, and I'm very happy that I'm sharing one of my PhD project, which is a collaboration with Moritz Small Group at the KFZ, and it's about the active repressing of alternative cell fate, safeguard hepatocyte identity, and prevent liver tumorigenesis. I'm gonna start with the sulfate plasticity, and from the Wellington landscape, we know that the cells start from the stem cells going toward the mature cells, or also we can reprogram the cells going toward the pluripotent stem cell, or even directly reprogram. And these all can happen with some biological process, such as activating this master regulator like ASCL1 from the fibroblast toward neuron or MyoD1 from the fibroblast toward the myocyte. But then what ensures the sulfate fidelity? At the same time, there are other group of transcription factors in addition to these terminal selectors that limits alternative fates or block the dedifferentiation and in general safeguard the cell identity. And such a transcription factor has been shown as a MIT1L, for example, which is the safeguard for the neurons. It blocks the other cell fates and then help us to have a better neuron. And then if we look at it in general as systematically, these terminal selectors are like ASCL1 are like a keys to this safe. And then if they are collaborate with the repressive safeguards like MIT1L to ensure the cell fate fidelity. And then so this brings us to a couple of questions. First, do other cell types have such a specific safeguards? And also, can the prevention of the unwanted plasticity blocks any disease? So this motivates us to start to looking for such a transcription factors, and then they have a couple of features such that they should have a cell type specific expression, they should repress unwanted cell identity, and enhance the desired cell fate. We start to finding this transcription factor with implementing a computational screening, which is based on the three steps. In the first steps, we are looking at the TF expression, and we are expecting that TF expression should be higher in the target cell type and it should be low in the other cell types. And then this is, for example, the case for MIT1L that it's very high in a neuron and it's low in the other cell fate. And in the second step, we are going to look at its target. We are expecting its target to be on the other cell types and not in our target cell type. So it should be depleted there and then enriched in other cell fate. And then as you can see in this example for MIT1L, it's depleted in neuron and it's lowest among the others and it's enriched into the other. And if we, in the third step, we put these two factors together, we can actually calculate the Z-score and then identify the potential safeguard candidate for our target cell type. So, and this is the general approach. So we can actually apply it on other cell types, not only neurons. And you can see here, we are working with 18 cell types from all the germ layers. And here we can start to focusing more on one of the cell types. And because we already work with them, we already know about the neurons, we focusing more in another germ layer and more specifically on hepatocyte here. We have 6 candidates for hepatocyte. And then we start to narrowing down to find one potential safeguard candidate, and then we filter them based on their lifeline expression, and also we design an experiment to reprogram MEF toward the hepatocyte, and then adding this transcription factor 1 by 1 to the 4-in-1 culture, which is supposed to have the hepatocyte, and see which one works better. And as a result, you can see here that the PROX1 dominate the other one in terms of number of the hepatocyte cells and also in terms of the expression of albumin. So PROX1 could be an safeguard for the liver fate, potential safeguard for the liver fate. So now I want to back to my second question and see, okay, if these safeguard can help us to prevent disease that are related to sulfate plasticity. And one of these diseases, of course, hepatocellular carcinoma. And the first thing that we observe is that in a normal versus hepatocellular carcinoma, we have a much higher expression of PROX1 in the normal, significantly higher in the normal. And then the second thing is that we saw that the survival rate for when we have the high PROX1 expression is significantly higher when we have a low PROX1 expression. And these two motivate us to actually look at the PROX1 overexpression in vivo in the cancer model. And we observe that actually when you overexpress the PROX1, there are no tumor formation. So and this also lead to, of course, a significantly higher survival rate. So it shows if you overexpress the PROX1 will act as a tumor suppressor and help us to prevent the cell plasticity in hepatocellular carcinoma. So this all motivates us to start to dig more and try to understand the mechanism of the PROX1 and try to understand how PROX1 ensures the sulfate fidelity. For that, we designed this time a single cell experiment that we overexpress PROX1 and then harvest the cells at day 2, 7, and 14. And by that, of course, we have the two conditions, which is the GFP and as a control, and also we have the PROX1 overexpression. The first thing we have done, we tried to identify the hepatocyte cluster, so the cells that are not confused and went toward the hepatocyte, and you can see these are the blue cluster here. So first, we want to look at these hepatocyte cluster and let's see, okay, how behave the cells with the PROX1 overexpression versus GFP. We can see at the day 2, 7, and 14, all the cells with the PROX1 overexpression, which are the orange here compared to the green, has a higher hepatocyte identity. It means there's a higher hepatocyte marker genes expression. And at the same time, what about the host identity? If we look at the MEF identity, which we start from, we can see at the day, again, at the day 2, 7, and 14, we have a lower expression of the MEF marker genes in the PROX1 compared to the GFP. So PROX1 help us to have a better and more hepatocyte and makes the whole reprogramming more efficient, and then at the same time it blocks the host identity better than the time the control. But then what about the alternative cell fate? Is it PROX1 useful to block the alternative fates? For that, we need to have a regulon for the PROX1, which is a set of target genes of a transcription factor. So for that, we designed an experiment, the cut and run experiment, and then with using cut and run, we can have a proper regulon for PROX1. And the next step is if you look at this regulon activity and compare it with the other cell fate, for example, here I'm showing the fibroblasts, you can see a negative correlation. It means that whenever the PROX1 is more active, the regulon is more active, we have a lower identity of the other cell fate. And then we tested it on the all 18 other cell fate that we had, and we can see that PROX1 has a negative correlation with almost all of them except hepatocyte, and it means that the PROX1 is silencing many but one desired cell fate. So when we see that the PROX1 actually repressing the other cell fate, we start to think, okay, we need to understand what are the important targets that PROX1 is using for repressing other cell fate. And for that, we designed another experiment, which we had the bulk ataxic data for day 2, comparing control versus the PROX1 overexpression, and also we had the RNA-seq data from day 7, 14, and 28, comparing overexpression of PROX1 versus GFP. And then this is the heat map for differentially expressed genes. We clustered them into the 4 clusters. And the first thing that we wanted to look is that if the PROX1 regulon is in, where are the PROX1 targets? And you can see PROX1 targets are enriched actually in cluster 3 and 4, which are the genes that got downregulated. They are lower in the PROX1 condition, where PROX1 overexpression versus GFP. And also, if you look at the regulon of 4 and 1, they are higher in cluster 1, which are the genes that are higher in PROX1 overexpression versus GFP. And but then what are these genes? We first look at the marker genes from these 18 cell fates and see if they are enriched in one of these clusters. And then interestingly, we saw that the hepatocyte and some hepatocyte like cells like cholangiocyte are enriched in the cluster 1, which are the genes that got upregulated in proximal overexpression. And then on the other hand, most of the other cell fates are ended up in cluster 3 and 4, which are the genes that proximal will target them and repress them. So now when we have all of this information about these marker genes, then we start to investigate it a little bit more. And for investigating more, we use the tools that we implemented in our lab, like called Granny and Grandpa, and more specifically Grandpa, which help us to find out about the important transcription factor that drives the differential expression. And then we use this tool to understand what are the important transcription factor that drives the difference between day 2 from the GFP versus the proximal overexpression, or at day 7, 14, and 28. And then interestingly, we see at the day 2, PROX1 is the only important transcription factor, which it makes sense. But starting from day 7, we start to, beside the PROX1, we start to see the other transcription factor, like PRX1, EBF2, OSR1 at day 14, and or at day 28, ELK3. And then the first thing, if you look at the target binding of these transcription factors, they are pretty much similar to the PROX1. They are only binding at the cluster 3 and 4, which was, if you remember, was the responsible for the genes that got downregulated in PROX1 compared to GFP, which means they are getting repressed. But if we're looking at the activity to TF expression compared with the Regulon expression, it means most of them has a positive correlation. It means they are activator, and then it means that we can end up with this cartoon that the PROX1 repress activators like a PRX1, PPAR gamma, and EBF2, and toward the, repressing these activator, PROX1 can block the fibroblast program and adipocyte program. So after we have this candidate, now we want to confirm their role in hepatocyte. So we design an experiment for overexpressing PROX1 or PPAR gamma, and at the same time also knocked out of the PROX1 and PPAR gamma. And then what we observed is that in case of the overexpression, we have a lower relative albumin protein, which means when they are when they overexpress them in a hepatocyte, we have a worse efficiency. But at the same time, if you knock them out, then you have a better efficiency. You have a relative higher albumin protein. That means that actually, indeed, PROX1 will help to help the efficiency of the reprogramming with blocking these two transcription factors. So I want to now, when we know the mechanism, or we want to check if this mechanism is also applied to disease like a cancer. So in this time in human, we look at from the TCGA data, we look at the correlation between the PROX1 expression in HCC and compare it with the PRX1 and PPAR gamma. And you can see in the first two lines, they have significant and high negative correlation. And then we thought, okay, so what about the other cell types that are very close to hepatocyte, like a cholangiocyte? We see that, okay, it also has a negative correlation with KRT19, which is one of the markers for cholangiocyte. And at the same time, it has a positive correlation with HNF4A. So this, the whole system can also be applied to cancer. And now we ask this question from ourselves, that what would be happen if we knock out the PROX1? What would be happen in the cancer? And then the first thing that happened is when we knock out the PROX1, the first thing that we observe is that the size of the tumors are much smaller in control than compared to when we knock out the PROX1. And then the one interesting observation was that when we look at these GFP positive tumors, we saw that in a stage, for example, in this top bar plot, we have a lot of cells with the high HNF4A expression, a lot of GFP positive tumors with the high HNF4 HNF4 expression. But then when we knock out the PROX1, we saw that the 80% of them are now has a low expression. And at the same time, in control case, we have a lot, 98% of them with KRT19 low expression. But when we knock out the PROX1, we had around 57% with high KRT19 expression. And it means that actually when we knock out the PROX1, we have a transdifferentiation from the hepatocellular carcinoma into cholangiocyte carcinoma. And if you look at the marker genes in the heat map in the right, you can see that the markers of the hepatocyte are going down when we have the SH-PROX1, and the cholangiocyte markers are going up. And then this all can give us this idea that PROX1 actually blocks transdifferentiation of the hepatocellular carcinoma. To sum up and a conclusion, the one statement would be that PROX1 safeguard the liver fate. And as we had from the MIT1L in neuron, we can somehow have the PROX1 in hepatocyte, and then PROX1 induce and maintain liver fate in vitro and in vivo. PROX1 repress the non-hepatic cell fates, and at the end, PROX1 can regulate liver tumor identity and its progression. So by that, I want to thank you all for listening to my presentation, and shout out to Moritz and Bryce, awesome collaboration with them at the COPSET, and thanks for the listening.